Welcome to video 12 of Genetics the Code Broken. In this video, we're going to be looking at cloning of both the gene and whole organisms, and then you're going to, this is actually part of your assessment task, so make sure that you use some of these to help your understanding, but also use this as a platform to guide further research. So first we start off with gene cloning, and that's where we are producing just a specific gene that we want to copy, and a clone is an exact genetic copy. So by using genetic engineering techniques, we're able to produce unlimited numbers of identical copies of genes. And we say that when we do make multiple copies of these genes, we say that it's meant to that it's said to be cloned or amplified. PCR, which we've mentioned before, it's the best technique for gene cloning, and this allows it occurs outside the cells, and it amplifies a specific DNA sequence by multiple cycles of replication followed by heat separation and purification by gel electrophoresis. So here we have just, I guess, a quick infographic on how this PCR works, and we can see that we are in fact producing many copies of the same section of DNA, and that's that section that scientists have selected for their specific purpose. So what are some of the uses of gene cloning? Well, we produce multiple copies of a gene, which allows us to analyze them and research them in greater detail. One example of its use is in forensic investigations, where we want to actually do lots of testing on a small sample of the DNA which has been recovered. It allows us to produce protein products, hormones, vitamins, and antibodies for use. So some examples are human growth hormone and insulin, and it allows us to produce transgenic organisms with specific characteristics, such as those which we've discussed in our hybridization and our selective breeding documents. And then we can also have whole organism cloning, and this is a method of reproduction that results in offspring that are genetically identical to that of the parent organism. So by plants, this occurs by asexual reproduction. We can have artificial plant cloning, so that's by grafting, taking cuttings and tissue culture. And then we have animal cloning, where we see nuclear transfer technology. So how do both these types of cloning work? So what is the process? So I guess in gene cloning, we're using recombinant DNA technologies, and allows gene and genome sequencing to be done rapidly and effectively. When we talk about whole organism cloning in plants, we're talking about them being cloned from tissue culture. So these cells are incubated and grown on a medium, and they grow into whole plants. The offspring are likely to be identical to the parent plant, and it's a cheap and easy process. In animals, the process which we use is nuclear transfer technology, and that's where the entire genome from a cell of a parent is inserted into an egg cell that has had its nucleus removed, and then it is then implanted into the uterus of a mother and allowed to develop normally. This is, however, a slow, expensive, and inefficient process, and usually resulting in offspring that have shorter lives and poorer health than offspring that would have been produced normally. What are the products of these types of cloning? So in genes, we see the multiple copies of the one gene, and then this gene can be used for insertion into another organism or to create a particular protein coded for by that gene. In whole organism cloning, we produce a consistent population of offspring that are genetically identical to the original parent. The offspring, however, may not end up to appear exactly identical because of the effects of the environment on the phenotype, even though their genotype is the same. What are some of the benefits of cloning? So in agriculture, we see animals that have required characteristics, allowing them to produce more quickly than by traditional breeding techniques. We can use cloned animals to test new drugs and treatment strategies, and by using these clones, we can ensure that their responses to the drugs are more consistent, And one of the examples is using a cloned pig as a potential organ donor. Therapeutic cloning is the production of cloned human cells for use in research on human diseases. Stem cells, in fact, can be cloned and then triggered to develop into specific tissues and organs that are used to replace diseases and damaged ones. So, how can we work out is it a clone? And that one is by DNA fingerprinting, and that's the use of restriction enzymes to cut the DNA at specific points, and then we go through the process of electrophoresis, and we can see how similar sections of DNA are. And then the other is the DNA hybridization, where we allow the DNA samples to separate and then we cool it down and allow them to then join back together. And their degree of bonding reflects their similarity. And the more heat that's required to denature these DNA strands means that there's a greater similarity in the bases. So that's video 11. Make sure you tune in for video 12. That was video 12. Make sure you tune in for video 13.